Okay, well, I'd also like to start by thanking the organizers for inviting me to this beautiful place. It's been a great conference, and I look forward to sharing with you some of the work that we have been doing. Now, I have to also apologize because nothing about my talk has anything to do with integrative structural biology. What I will be talking about, though, is nucleic acids, which we haven't heard too much about, and I'll also be talking about dynamics as opposed to just structures. Now, let me start off by making a provocative statement, which is that no matter how many structures you solve in different functional states of a biomolecule, these structures are not predictive of biological activity. What I mean is you cannot take a collection of structures and from the structures determine, for example, the K-cat value of an enzyme or the binding affinity of a particular ligand to a protein or an RNA. Instead, these structures, by and large, are descriptive. We can't take them and really make quantitative predictions about the activities of the molecules. In order to be able to advance structural biology to the point that it's actually predictive, in my opinion, the way to do it requires, in fact, information about dynamics. So it's not sufficient to have the structures of a particular molecule in different states. You also need the kinetic rates with which these structures interconvert and their relative stability, so thermodynamics and kinetics. Now, what I'm going to be talking to you today is such an example where in DNA, dynamics of base pairs dictates the frequency with which errors occur during replication. So here's an example where the rate at which two structures interconvert determines something we all care a lot about deeply, which is the rate at which you make an error during replication, which in turn determines the mutation rate, which is fundamental, of course, to life. Now, let me start off by acknowledging the students who did the work. This is work carried out by two graduate students in the lab, Isaac Kinsey and Eric Smansky, and this is in collaboration with the laboratory of Zukai Su at Ohio State University. Now, as we all know, DNA replication is a remarkable process that is highly accurate and highly fast. And so there are three steps involved. The first step is the initial selection of the NTP. You have the polymerase, and it's selecting the NTP for interaction. And this particular step contributes a fidelity of 10 to the minus 4 to 10 to the minus 5. So normally, if you have a G, for example, that's being replicated, the polymerase will almost always pair it up correctly with a C. But once in 100,000 times, it will make an error and, for example, misincorporate a T, so pair a G with a T. Once that error occurs, it's not the end of the world because it's, of course, proofreading. So many polymerases have a proofreading functionality that allows them to correct mistakes. And in general, only 1 in 100 mistakes evade this proofreading checkpoint. And if, again, a mistake makes it through proofreading, finally, we have the mismatch repair system, which are damage repair enzymes, which can find mistakes in DNA and correct them. And if you multiply these three different stages, what you end up with something like 10 to the minus 10 overall fidelity of replication. Now, today what I'm going to be focusing on is this initial step, which really accounts for the most significant part of the fidelity. And the question that I'll try to answer is, why is it that polymerases make mistakes misincorporating a GT rather than a GC with this frequency of 1 in 10,000 or 1 in 100,000? Where does that number come from? Why is it 1 in a million? Why is it 1 in 10? And in particular, we're going to be interested in GT because those are the most common types of errors that occur during replication, roughly 10 times higher than, for example, GG or GA. Now, what's really interesting is if you ask the question, how often does an RNA polymerase make a mistake during transcription and misincorporate a GU? Or you could also ask, well, how often does a ribosome screw up and misincorporate a GU at the first or the second codon position? The answer is, in general, the probabilities are very remarkably similar. So across three vastly different structures, ribosomes, RNA polymerases, and DNA polymerases, these errors in misincorporation appear to fall within roughly 1 order of magnitude. And so one might wonder, how is this accomplished? And as I'll show you, it's all in the nucleic acid, and these other fancy ribosomes and polymerases really are just passively sitting around. Now, of course, we know now how replication, high fidelity replication is accomplished. It's accomplished by the fact that Watson and Crick base pairs have a very special geometry, which I'll refer to as the Watson and Crick geometry, highlighted by this blue rectangle. If you take GC, CG, AT, or TA, they all basically form the exact same shape, and that's what allows you to produce a structure which has arbitrary sequence and is extraordinarily regular. Now, of course, the polymerases have built active sites that are contoured precisely to the WatsonCrick geometry. So if you look at DNA polymerases, what you'll find is the active site has a shape complementarity that is perfectly matching to this WatsonCrick geometry. And this is how much of the fidelity is derived. So for example, if you were to introduce a GT mismatch here, this GT mismatch falls outside the blue box and sterically is therefore forbidden from falling in a catalytically active conformation within the polymerase. Likewise, if you took an AC mismatch, this would fall outside the confines of the blue rectangle. Well, then how do errors ever occur if they're sterically forbidden? Well, let's try to force the issue. Let's try to force these bases inside the box. What happens, of course, is you get a steric collision, and that surely is not acceptable. Now, Watson and Crick, in their second DNA paper, postulated that on rare occasions, if you were to form the enolic form of these bases, so this is enolic G here, where we've now moved this proton over here, now we can create a almost GC-looking thing that matches a Watson-Crick geometry. And likewise, you can take an AC and tautomerize the A to produce a Watson-Crick-looking AC mismatch. And these are the enol forms. And so they postulated, Watson and Crick postulated in their paper, that perhaps on rare occasions, these bases fall into these rare tautomeric forms, and that it might be the basis of mutations. This is back in 1953, which is pretty remarkable. Now, later on, Topolo and Fresco elaborated on this idea in the 70s, and what they said was perhaps not only is this the mechanism by which you get mutations, but maybe the energetics of forming the enolic state with this equilibrium constant of 10 to the minus 4 to 10 to the minus 5, maybe that also explains the frequency with which mutations occur. So in this model, if you look at the NTP pool, generally G's are all in their normal keto tautomeric forms, but occasionally there'll be a G in the enolic form, and that G can be misincorporated, and the probability of that occurring is roughly proportional to the concentration of this, which is given by this equilibrium constant, which happened to be, interestingly enough, 10 to the minus 4 to 10 to the minus 5, which is in fact the frequency with this misincorporation happens. Now later on, evidence for this model was provided for by these structural studies looking at these forms of damaged bases like O6-methylguanine. This is highly mutagenic, and it likes to pair with T, and structural studies showed that if you take O6-methylguanine and pair it with C, you get a mismatch, but it forms a Watson-Crick-like geometry when paired with T, explaining why this particular form of damage is highly mutagenic. So this was evidence that if you trap the G in a tautomeric form, you can actually get higher error rates. Now later on, studies by Goodman and others showed that if you increase the pH, the frequency of error, shown on the y-axis here, increases. And based upon that, it was proposed that maybe there's another species, not the tautomer, but rather an anionic species where we literally have removed the proton here from this T to create an anionic base. And this now can also form a Watson-Crick geometry. And the pKa for misincorporation was a 1 unit off from the pKa of this particular nitrogen. And if you were to then take variants of G, 5-bromo-U, 5-fluoro-U, what you saw was that the pKa shifted, and likewise the pKa of the NTP shifted, suggesting that maybe it is the anionic species that's contributing to errors, not the tautomer first predicted by Watson and Crick. And of course, all of these are Watson and Crick in geometry. Now later, crystallographic studies by the Kunkel lab, Lorena Bisi's lab, and Yusupova and Westhoff visualized these Watson and Crick-like geometries. This is a Watson and Crick GT, this is a Watson and Crick AC, this is a Watson and Crick GU, in the context of fully competent and active polymerases or ribosomes, showing that in fact these species can form inside the enzymes. However, because this is crystallography, we don't know where the protons are, so we don't know whether we have, for example, G-enol, where the G is enolic. Alternatively, you could have the TB enolic, or you could have the TB anionic, or the GB anionic. There are 4 possibilities for forming these Watson-Crick geometries, and because you don't see the protons, you can't discriminate between them, and so it's still unclear which of these species is contributing to these mutations. So we thought, let's try to use the most powerful technique that we haven't talked about so far in this conference, at least I don't think, which is NMR spectroscopy. And what I want to show you is that there are still many things that you cannot do without NMR. And so here in particular, we wondered, could we actually use NMR and characterize dynamic fluctuations of the GT from a mismatch wobble conformation to some of these exotic states, which we predicted would be low in abundance and short-lived, and perhaps measurable by NMR spectroscopy. And in particular, I want to address this question of where does this 10 to the minus 4 to 10 to the minus 5 come from. Now let me just give you a brief introduction to some of these NMR experiments. And what we have here are two signals, NMR signals emanating from a particular nucleus. And so if the nucleus is in or out, we get two different signals, and the height of these signals is roughly proportional to the population of those species. If those species now exchange on the micro to millisecond time scale, what ends up happening is we end up broadening the lines, and this B state, which is a low abundance, short-lived state, now has vanished from detection because this exchange causes broadening. And you can see, however, that the exchange process has not disappeared. It has left its imprint by broadening the resonance of the major A state. And so in the methodology I'll be talking to you about, called relaxation dispersion, what one does is look at this line width here and use it to learn about this exchange process, and ultimately learn a lot about this otherwise invisible B state, which has a very low population and a short-lived lifetime. Now, in the interest of time, I will go quickly through this, but essentially what we're going to do is measure the line width as a function of a power that we apply, and by doing that, we can back out these parameters of interest, the population of the A and the B state, the forward rate and the backward rate constants, and the chemical shifts of A and D, where the chemical shift carries a lot of information about the structure of the species B. Now, when we started these experiments, we were very hopeful, because when you take guanine and you deprotonate the guanine, so the nitrogen goes from being protonated to not protonated, this is associated with a huge change in the NMR chemical shift. That frequency changes by a huge amount, 57 ppm downfield shift. And likewise, if you deprotonate the uridine, you get a huge shift of 55 ppm. And since the NMR experiments grow as the square of this shift, we thought this would be a very powerful experiment to look at these conformational states. And so what we did was, we took a double helix and embedded a mismatch in there, and carried out experiments called relaxation dispersion experiments. And all I want to really show you is the data shown over here. This is data on the nitrogens, and what you can see is we clearly have a mountain, and the mountain has a particular frequency, which is the frequency of a state that is very low in abundance. This data is telling us that there is, in addition to the major ground state, wobble, there is an alternative, what we'll call excited state, which has a population of the tune of 0.1% and a lifetime of a few microseconds. And this alternative conformational state features a really downfield shifted nitrogen chemical shift for both the G and the T, exactly as would be expected if they were being deprotonated. Based on analysis that I don't have time to go over, we could show that what we were observing is the wobble converting into these postulated but never previously observed in the context of a solution state study, Watson-Crick-like tautomeric mismatches where you have the G in the tautomeric form or the T in the tautomeric form, 0.2% population, and the G being the dominant state compared to the T enolic state. When we increased the pH, we observed another species, and this species we could show was now the anionic T, which we could show forms at an astronomically small abundance of 0.001%. So think about what methodology could ever describe or measure a species that has this kind of abundance and that differs by merely less than an angstrom movement and just removal of a proton. This is the magic of NMR. So now if you look at this species now converts with that, and we can get that also from the NMR data. And so now we finally characterize those species. And so the next question is, well, what does this have to do with the misincorporation frequency? And what I want to draw your attention to is these numbers, 1 per second here. It takes about a second to form this. It takes about a second to form that. And this thing has a population of 0.1%. Keep that in mind. Now let me tell you about how misincorporation frequencies are measured. These are pre-steady state kinetic experiments where you take, for example, a primer G and you extend it with C and you measure the rate at which that extension occurs. Then you do it again for a G paired up with a T. Obviously the T is going to be slower. And then you fit this to get a K observed, and then you measure the concentration dependence of K observed, and you do a Michaelis-Menten type analysis to obtain K-pol and KD. K-pol is the maximum rate constant for nucleotide incorporation at infinite NTP concentration, and KD is an apparent dissociation constant. Now let me show you what we knew before our studies. What I told you is that the mistakes happen with frequency of 10 to the minus 4 to 10 to the minus 5. Now, of course you might think, well, the polymerase surely binds the GT with weaker affinity than the canonical GC, and that is true. It binds, the binding accounts for a factor of about 10 to the minus 1 and 10 to the minus 2 discrimination. So the polymerase already avoids mistakes to the tune of 10 to the minus 2 because it likes to bind GC better than it likes to bind GT. So where does the remaining 10 to the minus 3 come from? And the answer is it's the rate of misincorporation. So polymerases misincorporate the incorrect NTP 1,000 times slower than the correct NTP. So not only does it not love the GT as much as GC, it's slower at incorporating the T by a factor of 1,000. And look at this number, 1 per second. So we were struck by the fact that the rate with which the tautomeric species form, as measured by NMR, which is 1 per second, coincides precisely with the reduction in the catalytic rate for incorrect incorporation. And of course, this number is 10 to the minus 3, which is equating to the 0.1% population that we observed. So we constructed a model. We thought, okay, so here's a minimal model for correct incorporation. You have a G, in comes the C, you get binding, there's a conformational change, and then there's catalysis. We thought maybe for incorrect incorporation, it's basically the same thing, except you're binding a T to form a wobble, that's a weaker affinity event. And then we insist that there's a tautomerization step where the GT becomes a Watson-Crick-like species. And upon doing that, everything else proceeds just as if it were the correct GC. So the model assumes that once you form a Watson-Crick-like tautomeric species, the remaining steps in chemistry occur as if it were a perfect Watson-Crick GC base pair, because the polymerase can't tell the difference between a tautomeric GT and a Watson-Crick GC. Now, of course, we had measured, I showed you that we measured this step by NMR. We have all the rate constants for formation of these species, so we could plug the rate constants from NMR. All these other rates are available from the literature, and we could therefore predict what the misincorporation frequency will be under different models. In one model, we assume just the tautomer is able to evade the polymerase and make it in. In another model, it's the anion that makes it in. And in a third model, both of them make in. And so let's see which of these models predicts the data. So we're going to do the kinetic simulations and see. So here's now 3 different polymerases, Pol epsilon, Pol beta, and T7. Here's the error of frequency measured experimentally. And if you ask the question, how well does our model, where we assume both the tautomer and the anion are allowed to be misincorporated, you can see the data predicts the experiment quite nicely. If we assume it's only the tautomeric species, again, it works quite well because the anionic species is so low populated that at this pH it has a very small contribution. A model that assumes only the anion makes it clearly fails, so it's clear that the anion is not the species that's responsible for misincorporation. And of course, a model where you only assume binding also fails. Now, what about the pH dependence? Remember I told you that in the literature, there was data showing that if you increase the pH, you increase the misincorporation frequency. So clearly, it can't be the tautomer because the tautomer is pH independent. And if you take the model again that combines this, the tautomer and the anion, what you can see is we can explain that if you look at the pKa that we obtained from our model, it matches very nicely with the experimental pKa, both for regular DTTP and for 5-bromouridine. And so what we have here now is a model where, in fact, both species, the Watson-Crick-like tautomeric species, as well as the anionic species, are contributing to misincorporation. And so what happens at regular pH, most of the misincorporation proceeds through the tautomeric species, mostly because of the genolic species. So 99% of the errors are happening because of the tautomer at neutral pH. When you increase the pH, now you have a more significant flux going through this anionic species, explaining the pH dependence of misincorporation as this anionic species increases in population. For 5-bromouridine, which is a mutagenic form of T, what happens is that now most of the flux proceeds through the anionic species, even at regular pH, because of the depressed pKa of the 5-bromouridine. And if you increase the pH, then the flux proceeds even greater through this anionic species. Now, as I mentioned before, so essentially what I've been telling you is that the 10 to the minus 5 comes from 2 parts. One is the fact that the polymerase likes to bind GC better than GT by a factor of 10 to the minus 1 to 10 to the minus 2. And the remaining factor is the 10 to the minus 3 factor comes from the fact that the frequency with which the GT becomes a tautomer is roughly on the order of 10 to the minus 3. And as a consequence, since the polymerase enforces the Watson-Crick geometry, that is, in the end, that leads to the resultant 10 to the minus 5 misincorporation. Now, of course, as I said, these error rates are pretty similar across transcription and translation. And I just want to show you data here where we've now carried out the measurements for an RNA-DNA hybrid mimicking transcription, an RNA-RNA duplex mimicking translation. And what we see is, again, very similar misincorporation frequencies. So here, no matter what the nucleic acid context is, we're getting the same flickers of the mismatch into the Watson-Crick geometry, and we think this might be the clock that's dictating the mutation rates, and that the polymerases are sitting passively enforcing a Watson-Crick geometry, and when the nucleic acid slips into this Watson-Crick geometry, then the polymerase is fooled, or the ribosome is fooled, and they eat up and incorporate. Now, as a historical context, I received a letter from Jim Watson after this paper came out, and you might wonder, how did they even think about this tautomeric species back in the 1953? And the answer, it turns out, is that in fact, as many of you may know, Watson was using the wrong forms of the tautomeric species to begin with. So when they were trying to model build, initially, they were using the incorrect forms of the G and the T, and this was obtained from a particular reference here. There's a book. You would open the book and you'd see the G in the enonic state instead of the ketose state, and that is probably what led them to postulate the existence of these mutagenic species. Okay, now I'm just going to conclude by reinforcing the magic of NMR. So what I showed you today is that by NMR, one could measure a conformational state that has an abundance of 0.01%. That's what it looks like to be a molecule in the sea of excess 10,000. This is the abundance of the conformational states that we're seeing. I think it's pretty magical that you really can see this by NMR. But not only that, it's not only that this is low in abundance. Remember, it's also short-lived. So on top of this red dot is not there all the time. You don't have the lucky. If it was there all the time, it would be super easy. Let's make it harder. This red dot is in fact so short-lived that I can't even show it to you here. It goes faster. You have to imagine it towards a magnitude faster than that. So this little red dot is there in such low abundance for such a short period of time, and it merely involves movement of protons, which none of you can see, by cryo-EM and X-ray, and yet NMR can pick it up. All right, so with that, I'll finish by thanking again the people who did the work, and thank you for your attention.